All right, good afternoon. I know I'm a little late getting this up, but I wanted to kind of have a dry run with my live class last night, and it's a good thing I did. I found some mistakes. I'm going to do the best I can. Teaching math online is not ideal, but I hopefully I can get this concept across to you guys. So if you're on page, so again, I always suggest you have your lab book out and open and ready to go. So if you're on page 131, you'll see all the vocab words. If you've been following along with the lecture or the videos, those vocab words should all be familiar except for chi-square and degrees of freedom. And we're going to go through that today. So the first thing you would have done is you would have been sitting with your partner and you would have been asked to take a coin out and flip it 50 times. So everybody been flipping. I think most of us realize that when we flip a coin, we have a 50-50 chance of heads or tails. And this is all probability. But when we talk about probability, we're talking about it's a zero, it's not what you expected, it's not the heads that you called out, or it is the heads and that's one. So the probability is zero, it didn't happen, or one, it did happen. And so when we flip that coin, we know we have two sides of that coin, heads and tails. And so what's the possibility of that one outcome? It's 50-50. So when we did this, you guys flipped your coins, you had 27 and 23. So with that, what we would expect out of 50 tosses, we would have expected 25 heads and 25 tails, and we didn't get that. And so the question that we're going to ask in this lab is this a normal deviation from our expected, or is something else going on here? Is something influencing the results that we are getting? Now, what we would have done then is I would have put a chart up on the front board and you would have filled in your group's information and we would have had a class total. So I'm saying, all right, we had 5 groups and so our totals are 120 observed, 130 observed, that's 250. And so what's my 50% of my total 250 tosses? That's 125 and 125. So that is what you should be filling out for your table on page 133. Then you would be answering the questions. Okay, so the questions talk about, do your group's observed number fit the expected well enough to support the hypothesis? So that is the question. That is what we're asking. Is this close enough to support it? And again, these are kind of opinion questions, and that's where we're going to go, where we're going to use some mathematical analysis to say, yes, it supports my hypothesis. My hypothesis is that I will get a 50% heads, 50% tails, or no, it does not support my hypothesis. And then it asks, are the class totals closer to your hypothesis? Well, this again is not a question that I would grade, and I don't grade the in-lab stuff anyways, but I have times where the class total is much closer. And then I have another example where if your group got 25-25, you can't get any better than that. So it's really kind of a probability issue. Either way, what we would expect is that the more number of tosses, the closer we should get at our expected. You know, if you just toss it 10 times, you know, you might get 7 heads and 3 tails. But if you tossed it 100 times, it would be pretty close to 50-50. So what we would hopefully expect is as we increase our sample size, our numbers are more accurate. Now, they have this thing on page 134. I would highly suggest you highlighting this column like I have. This is our chi-square chart. Now, your chi-square chart is going to tell you what values are allowed. This is what we call our allowable chi-square chart. And so with that, what we are going to determine here is that we're going to say a 1 in 20 chance that something does not follow along our hypothesis is acceptable. And so that's a 0.05. So this is the column we're going to use. You will be given this chart on the lab practical and you'll be expected to know how to use it. Now, if, for instance, just off the cuff here, if you have a medication and there is a probability that the side effect could lead to death, which I did have that on one of my medications. I should have taken a picture of it and saved it and I put it up there. And I'm like, How is death a side effect? That's a final outcome. But anyways, let's just say that 1 out of 20 people die if you take that medication. That is not acceptable. So this is where our statisticians would say, well, you know, we want something even smaller. We're going to say we're only going to take a 0.99 deviation. And you can see how small our number is. All right. So for different purposes, we would use different columns, but for the purpose of bio 123, we're going to use a 0.05, 1 in 20 chance that we fall outside our expected numbers, observations, whatever you want to call it. Okay. So make sure you get that done. So I would highlight it. All right, so for procedure 10.2, if you read on past that chart in your lab book on page 135, it kind of explains what the chi-square value is, the chi-square test. It gives you an example. It fills that out as you go through. They talk about marbles. If you need some additional support, great thing to read through that. It kind of holds your hand through it as well. I'm just going to jump through and do this myself. So for procedure 10.2, what we're going to look at is we're going to look at your data here, your class data. So it asks you to use your class results in procedure 10.1. And so our hypothesis, again, very important. You need to know your hypothesis, because what we're doing here is what we call, we're supporting or we are falsifying your hypothesis, or a falsifiable hypothesis is also called a null hypothesis, N-U-L-L, null hypothesis. So with this chart, you're going to fill in, we said we had 120 and 130. We said our expected was 125. Ooh, I forgot my 2 there. So there should be a 2 in there. Let's put that in there. That'd be 250. 250, right? 250. All right. So then when we go ahead, you don't need to fill in this slot here. I wish we'd fill that in, but you don't need to worry about that box. Matter of fact, this box, you don't have to worry about that either. We actually put numbers in there. Okay. So, or letters to explain what this box is. All right. So you take your observed, and that's what this is telling you. Your observed minus your expected. My observed is 120, my expected is 125. So 120 minus 125 is -5. Now, then you're going to square it. So -5 times -5. Remember, when you square a negative, it goes positive, and that's 25. 130 over minus 125 is a +5, and 5 times 5, 25. For the next column, you're going to take this value, so our 25, and we're going to take our 25, and we're going to divide that by our 125. This is so awful doing it this way. This is why I said I'm glad I'm not a math teacher, but I do have it on the next chart that I scanned. So that gives me a value of 0.2. Same thing here, 25 divided by 125, 0.2. Now, when you get your calculated chi-square, what you're doing is you are adding this column. And so 0.2 plus 0.2 gives me 0.4. This is my calculated chi-square. All right, so this is my calculated chi-square value. Now that we have that calculated chi-square value, well, what the heck does that mean? So what we're going to do is we're going to say, well, what's our allowable? What are we allowing for this deviation? And what we look up, when we look this up in our table, we have this thing called, on the side, you have what's called your degrees freedom. Now, degrees freedom, okay? So that's your first question there. How many degrees of freedom are there? Well, when we go back to our table, notice here, outcome. We have 2 outcomes. You have heads or tails. So you have two possible outcomes. So when we figure out our degrees of freedom, you take your number of outcomes minus 1. So my number of outcomes was 2. So my 2 minus 1 gives me 1. So then I'm going to look up what is my critical chi-square value, and that is where I'm going to go to this table right here. So n, that's my degrees of freedom. My n was 1. So I'm going to go over to my 0.05, and so that means right here, my allowable chi-square is 3.841. All right, so that's my critical chi-square value or allowable chi-square value, okay? Critical meaning if we go above this, our hypothesis is null. It's no good. So is the hypothesis supported or falsified by the data? I put yes. Yes, it is supported. The calculated chi-square value is less than the allowable. So you could even put down that your calculated are 0.4 is less than 3.841. So what we're looking for is for you to use your data to support this statement. Why is it supported? Why is it falsifiable? All right, your next lab that you're going to do is now you're going to take 2 coins and flip them. And with flipping 2 coins, we have a couple different possibilities here. We have 2 heads. You could get both your coins come up heads. You could get 1 heads and tails, or you could get 2 tails. So you have to flip them at the same time. I have a lot of students who do not listen to that instruction and they get their flips and it's 100 flips and they're like, I'm like, you can't do it that way. You can't do 1 and 1. You have to do them together. So it's not a matter of one person can flip one coin 100 times and one person can flip another coin 100 times. No, you have to flip them together. Did you get 2 heads? Did you get 2 tails? Did you get a heads and tails? Okay, so once you flip them, and I have flipped them for you, I observed I have 21 heads, 56 heads, tails, and 23 tails. Now, my expected is 25, 50, 25, which gives me my 100. Many of you are going like, how in the world did she get that number? Well, how did I get that number? Well, if we do a Punnett square, here is our coin. So our coin, we can set this up like our coin has 2 possible gametes. It can have a tails, it can have a heads. Same with the other coin. Your 2 possibilities are tails, heads. When I put them together, I can get 2 tails here, 2 heads here, heads, tails, or tails, heads. It's still one of each, right? So I have 25% homozygous, we'll say this is dominant, 25% homozygous recessive, 50% heterozygous. Doesn't matter gene-wise if which one comes first, okay? You're going to show that dominant trait. So what I did was I looked at my percentages, 25% big T, 25% big H, 50% one of each. So therefore my ratio is a 1-to-1 phenotypic ratio. That's what I'm seeing. Okay, so when I look at this, let me go back. I'm going to, see if I can draw this. This is 25, right? So what's 25% of 100? 25. This is 50%. What is 50? Because I had 2 squares, right? What is 50% of 100? Well, that's 50. And again, I'm looking at my total. And then what is 2 tails? My probability of 2 tails was again 25%. Times that times 100. And we get 25. So that is how I get my expected. This one thing you need to know how to do is to get your expected, and we'll be working on that for this particular lab. Now notice my outcome. I have 3 possible outcomes here, okay? So when we do our math, I did my 21 minus 25, which is 4, which gives me 16. When I fill this out, just to save for punching buttons on my calculator, I just go all the way across. So I complete the whole column here. or should I say row? Sorry, rows across. So I go 21 minus 25 is 4. 4 times 4, if you don't know where your square button is, remember when we square something, we multiply it by itself. So 4 times 4, that's 16. And then I'm going to take my 16 minus my expected, right? I took my squared value, this column, over my E. So it's 16 divided by 25, and I got 0.64. Did the same here. 36 divided that by 50. I got 0.72. 23 minus 25 gives me -2, but when you square -2 times -2 is 4. 4 divided by 25 gives me 0.16. Now, to get my calculated chi-square, I add up this column. So that gives me 1.52. Okay. And then we're going to ask the same questions. How many degrees of freedom are there? Well, we had 3 possible outcomes. So 3 minus 1 gives me 2. Look in that table. When I look up in that table, my critical chi-square value, let's put that table up real quick. So my n is 2, so I'm going to go across here. So 5.991 is my critical chi-square value. Is your hypothesis supported or falsified? So again, yes, it's supported. All right, so I should have wrote, yes, it is supported. I just put yes. I was in a hurry. My brain was moving faster than my fingers could type. So yes, it is supported because 1.52 is less than 5.991. All right, so moving on to 10.4, now ask for your class data. So again, I'm going to say we had 5 groups. Here's our data. That's 500 tosses. To get my expected, I'm going to take, so again, when you get your expected, remember, you're not taking your observed, you're taking your total number. So what's 25% of 500? 125. What's half of 500? 250. 25% again, 125. That should add up to your 500. If it doesn't add up to what your total, if these two don't add up, then you've done your math wrong. Maybe you've taken your 25% times 120. That's not the way to do it. You always take your numbers here from your total, your percentages or your probability from your total. And then you're going to go ahead and you're going to fill out the rest of the table. Okay. So I do not fill out the rest of the table for you. That is something you need to check when I put the answers up. Some of these, you're actually copying some of these tables into your lab review for me to check. So you need to go ahead and figure out your calculated chi-square and then answer the questions. All right, then we're going to move on to red pigments in plants. And what we would have given you, it would be a container of pipe cleaners, red pipe cleaners and green pipe cleaners. Now, it is really important when you look at page 138, it says procedure 10.5, but the information that you need in that is in the paragraph above it, where it says monohybrid cross of the anthocyanin gene in sorghum plants. When you read that, it tells you which one is dominant, which is red, and which one is recessive, which is green. It also gives you your hypothesis. Your hypothesis is that you have a 3-to-1 ratio of red to green. So I'm going to give you these numbers right here. And again, I would try to do the math to make sure you get what I got for the expected. So I'm going to take, if I said 3-to-1, that's basically saying 3 squares in my Punnett square are going to be red and 1 is going to be green. That would be the same as 75% and 25%. So you're going to go 81 times 0.75, and that's going to give you your expected for your dominant. Then you're going to take your 81 times 0.25, and that's going to give you your expected for your recessive. And notice they do add up. Then you're going to go ahead and finish the table and get your calculated chi-square value and then answer the questions. The next one has to do with albinism in corn, and we are going to ask some conceptual questions about this, this situation here, because what happens is there is a mutation in the chlorophyll pigment. And because it's a mutation and the chlorophyll pigment doesn't get made properly, it can't do its job. These corn plants are literally white. And so, and it used to be a long time ago when I used to teach here, we would actually grow these corn plants for you to count, but now we just make pipe cleaners, green pipe cleaners and white pipe cleaners. But what we notice is that the corn plant will grow, about like this much, and then it's not able to make its own food. And because it can't make its own food, because chlorophyll pigment's not working properly, it dies. Right? So it basically has enough sugar in the seed that the mama plant packed in there for it. Because remember, plant seeds undergo cellular respiration under the soil. It's not until they get their leaves up and going that their food production line starts and they can, by photosynthesis, make their own sugar. But these particular plants can't make their own sugar at all, so they do die. So, it's kind of an important question when we understand genetics. Well, then how do you keep getting albino corn? And if you think about it, if we have a situation where we have green plants that are heterozygous, so if we do that, and this is what they're saying, if you read the paragraph above procedure 10.6, it talks about this. And so you're crossing two heterozygous corn plants. So if you need to do the Punnett square, again, you'll notice you'll get three. You'll actually get one that would be big A, which means it's homozygous dominant. You'd get two that are heterozygous. When we're talking about genotypes, that would be big A, little a, and then your bottom corner would be little a, little a. But when we look, so it'd be 1-to-1 ratio, which you should have those, pretty, if you've been doing your practice problems, that should be pretty clear. But our phenotypic ratio is what we see. And what we see is 3 out of 4 will be green. So that's our 3-to-1 ratio, 3 green to 1 white. And so that's our 75% and 25%. And so I'm telling you, you have 44 green and you have 17 white plants. This time you are expected to come up with the expected values. So again, this is where you need to make sure you know how to do this. Notice we have 2 outcomes. So it will be 2 minus 1 would be our degrees of freedom. The next lab, 10.7, that we would be doing would be our corn. And if you watch the videos with Marcia Hay, she showed you the corn. Okay, so with that corn, you're going to be counting, you would have been counting how many kernels are black, how many kernels are yellow. So I have given that number to you. Again, when you read the information, it tells you in the hypothesis that it's a 3-to-1 ratio as well. So based on all that information, you should be able to calculate your chi-square value. Now, the last part of this lab has to do with dihybrid crosses. So hopefully you have at least studied your dihybrid crosses before you get to this part. Maybe if you haven't, I would pause it and then go do the dihybrid crosses and make sure you have a pretty good understanding on how to do those, and then come back to this lab. So the first procedure, 10.8, they actually have the observed for you. You need to figure out the expected. Now, again, we're saying that this was a hybrid, a heterozygous or hybrid, right? 2 hybrid corn plants. So when we look at them, what we're saying is that they would be, you could say, big Y, little y, big S. So we had 4 possible gametes for mom and dad. So that's why we are going to have our big Punnett square, and that big Punnett square is going to give you a phenotypic ratio of our 9:3:3:1. So that is our hypothesis, that our kernels should be representing themselves in a 9. 9 would be the double dominant. In this case, for our P's, it would be yellow and smooth. 3 would be our recombinants. We got our 2 recombinants here, one dominant, one recessive, one dominant. So green smooth, yellow wrinkled. And then our poor little guy here, our double recessive or homozygous recessive, that would be green wrinkled. So we would have those 16 squares. So a 9 out of the 16 would be double dominant. 3 out of 16 would be one recessive, one dominant. 3 out of 16 would be one dominant, one recessive. 1 out of 16 would be recessive. This is what we're going to use our 556 to get our expected. So here, what I did was I took my 556 times 916. If you're not sure how to do that on your calculator, I do it by going 9 divided by 16, and then I hit the equal sign, and then I multiply it times 556. That gives me 312. When it's usually in like 0.75, 0.5, or the quarters, you could say, I usually keep those. Now, I do understand there will be some rounding differences, so if your number's not exact, but it's close, you're good. So then for our 2 recombinants, that was 3/16. So you take your 556 times 3/16, and that gives me 104.25. So remember, I have 2 of those. And then my double recessive, 556 times 1/16, and that gives me 34.75. So that is how I got those numbers there. Now I do add them up. They should add up to 556, which they do. They do add up. And then you're going to do your math. So then you can check your math with what I have here. You should be right around this number, 0.516. Like I said, you may not be exact because of rounding differences, but you should be pretty close. Now, the last one you actually do, so 10.9, you are actually going to do the counting. So you would be given another ear of corn, and you would have to count, and you can't just count black and yellow or smooth and wrinkled. You have to count them together. So how many kernels are black and smooth? How many kernels are black and wrinkled? How many are yellow and smooth, yellow and wrinkled? So you need to count all those. And again, it is our expected ratio is 9.331. And so this time you are going to calculate this out. So you're going to, and that is the last part of the lab. So you're going to calculate that out, answer the questions, and then you're going to do your lab review. So for your lab review, you're going to answer all the questions. And then if you notice, this one's going to be a longer one. So on page 146 and 147, they're asking you to kind of transfer that information from your lab over to the lab review. And in fact, and then they have a practice problem for you, which is kind of interesting because I've got rounding differences on my 2 tables, but that's okay. They're very close. All right, so make sure you get those completed and answer the questions and turn in on Friday. All right. So again, if you guys have questions, I know that some of you guys I've never heard from or whatnot, but please feel free to shoot me an email. If we need to Zoom, you know, I am more than willing to do that whenever you guys need to get that extra support. All right. I will talk to you soon. Bye.